Hi, this is the Team 5 and this is our big data project. So the topic of the project is genomics drug response prediction. So what we're trying to do in this project is we are trying to predict the drug response on basis of gene expression. When we were choosing this project, we wanted to choose a field that required big data for its very existence. And on a bit of research, we got to know that genomics is that up and coming biotechnology field which requires big data. The reason is that uh, there are facilities which analyze genes on the basis of chromosomes, DNA code, etc. to formulate a single metric called as gene expression, and this expression defines the behavior of the genes. That means rather than making data set of different patients on the basis of their uh, body, BMI, and the location, etc., we can generalize patients on basis of their gene expression itself. But the issue is that these facilities generate a huge amount of data. It can be almost 3 GB per genome, and if you take into the consideration how many genomes are processed in a day, it can be more than 6 GB of data per day, which is huge, and it of course requires big data. So we are just working on a small quotient of this genome benchmarking project. All we are trying to do is trying to predict the drug response based on genes. So let me acquaint you with the data. We are working on basically four data sets. I'll open them up. We have the gene metadata, we have the gene expression omnibus, we have the gene ontology, and we have the patient metadata. So the patient metadata contains just the general information of the patients with two specific fields. One is the disease and the other is the drug response. The other is the gene metadata, which contains the ID of the genes, the target of the genes, position, etc., and the function of the gene. Then we have the gene ontology, which actually contains of more deeper information about the data, which is actually some biotechnology specific terms, and and the last value is the most important. We have the gene expression omnibus. This data uh, contains a set of the genes, the patient ID related with the genes, and the expression value of these genes. So let's talk about the process of how this project goes forward. Okay. So what we are doing is once we import all the files uh, in our Spark, uh, we are doing one simple thing. What we do is we first take the gene omnibus data, which is right here, and we try to join it with the patient metadata and the gene metadata. As you can see here that we have this genome omnibus data, and we have several gene IDs in here, and all these gene IDs are also linked to the patient IDs right here, and each and every of this gene ID and patient ID pair has their expression values. So in order to create a greater data set from this, what we are doing is we try to pivot the data on the gene ID such that this gene ID, rather than being a column itself, all the individual genes are transformed into individual columns, which you can see right here. You can see I have the patient ID, disease, drug response, and we have all these genes and their expression values in the form of a pivot. What is the use of this? So if you can clearly see, if I can take all these genes from 100 to 1000, all these genes, and I try to relate it with my drug response, I will get a kind of regression uh, analysis which can relate all these gene types to this drug response. And if I have substantial amount of data, I mean, because any human being will not, of course, be tested on just one gene. I mean, he will be tested on around thousands of genes. So if we have all those thousands of genes and we try to link that to the drug response of that individual patient, we can easily be able to identify the other patients who have similar genomics, and we can also predict what kind of response would they give to some specific drugs, and that would really simplify the drug testing process in the medical industry. As you can see, this field is not new. This has been there since 2007, I mean, just near the advent of big data. And this is one of the maximum sourcing big data technologies. Uh, uh, Going forward, you can see this is a white table. I mean, you can see that my gene ID start from 1 and they are going up to 999. This is a white column, and right now we have just taken a small sample because we have just 1000 rows, which is not optimal because if you see the actual real world data, it contains around uh, 10 10 raised to I mean, in the order of 10 raised to 10 rows, which is something which can really make the models learn because of the sheer quantity of data. Uh, Going below, we have uh, looking into this data, we narrowed down that we would go along three approaches, which seemed very potent for this kind of analysis. So we narrowed down our approaches to linear regression, decision tree, and random forest regressor. I mean, actually, I've spent a lot of time just to define the data, but I think it's very important that we all can get a sense of what the data is actually speaking.
We have done some exploratory data analysis to get an idea about our data. So first we checked the data for null values. So as you can see, out of all the columns, there is no null value present in our data. So a reason for that is we have used a synthetic data generator provided by the National Center of Biotechnology Information. So due to that, we have synthetically generated all of the rows in our data. So because of that, there are no null values in data because we have already handled that before loading the data. So let's move on. And so, so first we are checking the count of untargeted and the targeted genes on data. So as you can see from this, the untargeted genes and the targeted genes are pretty similar, which is really important because we have a lot of genomic data in our data set. And to get a good prediction, we need to see that the targeted and the untargeted genes are similar. So as the both of them are pretty close, so we can go ahead with the analysis. So next is the age distribution. So from this we just wanted to see the demography of our data. So as you can see here, age 24 has the most number of count. So this gives an overall idea about the age of the patients. So next is the age distribution among the patients. And after that we are seeing the gender. So we have taken the count of the gender from the patient's table here. So we can see that the our data is basically equally divided into patient into that both of the genders. So we have 51% males and 49% females. So so next is the drug response versus the age and the gender distribution. So from the graph, we can see that the drug response is equally divided into all of the ages, and the distribution is we can see that the distribution is also equally divided between males and females. So after that, we just check the count of number of patients in each disease. So we have our diseases numbered from 1 to 20, and here you can see all of the diseases have counts of around 50. So these are the counts of the diseases. So next we decided to find the correlation between the expression values of all the genes and identify the genes which have similar expression patterns and the ones which have opposite expression patterns. So to do that, we plotted the correlation between all of the genes, and this is the correlation matrix which we got. So as you can see, the number of columns in our data is close to 1,000. So because of that, the correlation matrix is not that great, to be honest. But on an average, we can see all of the correlations are in green. So that means that the correlation is more than 0 0.5 for all of the genes. So by using this correlation matrix, we narrowed down 319 columns which were really correlated with the target. So next is the modeling part in our project. So we have used three models. The first one is the linear regression. So while performing the linear regression, here we have given all of the input columns, which are the gene expressions, and after that we have created a pipeline, and the target variable is the drug response which we want to predict. So once we fitted the model, these are the results which we got. So you can see the drug response and the corresponding prediction for each of the patient IDs. And when we evaluated the model, these are the residuals which we got from the linear regression analysis for each of the patients. And after that, we can see the RMSC for this linear regression model is 26.10, and the R square value is around 17%. So this is the first model which we built. The second is the decision tree regressor. And in the decision tree regressor, we basically followed the same steps as we did in the previous model. And for that, we have got an RMSC of 33 and an R square of 14%. So after that, we went on to build another model, which is the random forest regressor. So in the random forest regressor, when we evaluated it, we got a better RMSC of 30, and the R square has also increased to 26. So according to the comparison of the three models, we can see that the random forest regressor gave us the best results. So after seeing the models, coming to the inferences of our project, so you saw that the predicted response rate for few of the patient IDs was really close to the actual drug response. So what that means is our new model which we have built will be able to predict the drug response for any new incoming patient whose gene expression history we already know. So if a patient comes in to a facility and we know the his gene expression history, our new model can predict if the drug response to that patient will be good or bad on a scale of 1 to 100. But when we evaluated the model, coming to the accuracy of the model, we can see the model is just the best accuracy which we got was 26%, which is not the best. The main reason for that is we have only taken 1,000 rows, and in the real world genomics benchmarking, the number of rows goes up to 10 raised to 5 to 10 raised to 10, which is really high. So given the infrastructure constraints, we could not get a really good accuracy for our model. But if we train our model on more data and we increase the size of the data, our model will be able to predict really better. So, so basically, this was our project, and we were successfully able to build a regression model which will predict the drug response according to the gene expressions. Thank you for watching the video.